Welcome everybody to the University of Melbourne Centre for Cancer Research seminar, Why are more people getting colorectal cancer? I'm Mark Jenkins and I'm the Director of Epidemiology and Biostatistics at the School for Population and Global Health. Today's seminar will be a two-player event starting with me, but soon passing over to our other presenter today, Associate Professor Dan Buchanan. We're both thrilled to be presenting in June as it is National Bowel Cancer Awareness Month. Bowel cancer, another name for colorectal cancer, does not have the high profile of breast cancer and prostate cancer. And we know that increasing awareness is one of the best ways to increase prevention and early detection. I'm pleased to say that the NHMRC recognises the significance of bowel cancer research as Dan and I, as well as our colleagues Onco and Professor John Emery, have all received investigator grants this round for bowel cancer research, which is terrific. Before we get going, I just want to let you know that there will be an opportunity at the end of the presentations for questions, but you need to enter them by using the Q&A button at the bottom of your screen. We are meeting today on the lands of the Wurundjeri people and acknowledge them as the traditional owners and ongoing custodians of these lands. We would like to pay our respects to their elders, past, present and emerging, and Aboriginal elders of other communities who may be here today. Welcome. Welcome to the UMCCR seminar. Okay, so in Australia, when we look at the entire population of all ages combined, there has been a gradual decrease in colorectal cancer incidence since 1999, a decrease of about 15 to 20% over 20 years. Note that colorectal cancer includes colon and rectal cancer combined. This decrease is probably due to screening. The National Bowel Cancer Screening began in 1996 and has been increasing in its rollout for all the age groups over that time. And colorectal cancer screening can reduce the incidence as well as mortality because it can detect the precancerous lesions or polyps and have them removed before they become cancer. But pushing against this effectiveness of screening are any increases that are happening across the population for colorectal cancer that might be occurring. So it's a fight, basically, a battle of prevention versus causation. And it's pleasing to see that the prevention is winning. Again, though, this is the picture for all ages combined. And it's a very different picture when we look at younger Australians. So these are graphs that I've made using the very latest data from the Australian Institute of Health and Welfare. And it shows that the incidence of colorectal cancer for different age groups of Australians. And they show that the younger the age group, so if we look at the groups in say the 20 to 24-year-olds and the 25 to 29-year-olds and 30 to 34-year-olds, the increase is much greater than people 40 to 44 and 45 to 49. Just a point on scale, these graphs have all been auto-scaled on the y-axis. So you'll note that for the 45 to 49-year-olds, the incidence rate in 2020 was approximately 10 times that of the rate in the 20 to 24-year-olds. But the point is here that the increase with time has been much more rapid in the younger the Australians are. What I haven't shown you are separate graphs for colon and rectal cancer. These are colorectal cancer combined. The increased incidence over time is even greater for rectal cancer. So these are all groups that are not having screening, very little screening because the national program doesn't extend to people under 50. These have also been, increases have been observed in USA and other countries. So it's not an Australia-centric issue, it's happening around the world. Theories for this increase? Well, there are several theories that have been proposed, and I'll go through them in a second. But in short, the reason for this increase is not known. We do not know why this increase has been occurring. The theories include Firstly, that there has been an increase in the prevalence of standard risk factors for colorectal cancer. And we know these also are risk factors for colorectal cancer for young people. For example, has the recent increase in obesity contributed to the increase in young onset colorectal cancer? There's been an increase in physical inactivity, which we know is a risk factor. There's been an increase in alcohol use in some teens, and we know there's been a worsening diet, which can be a risk factor for bowel cancer. One theory is, quite a novel theory, is that perhaps the decreased aspirin use in childhood, aspirin's no longer recommended for children who have a fever, maybe that decreased aspirin use has resulted in a loss of long-term protection, because we know that aspirin can provide a long-term protection for bowel cancer. Another theory is that there has been recently, this is an interesting theory, proposed by a world-leading gastroenterologist, that maybe there's been a deferring of defecation or a holding on, resulting in a longer transit time and therefore longer exposure of the fecal matter to the rectal wall. And maybe this has increased mutagenesis by some field effect, and there's some evidence it may be affecting KRAS mutations. Another theory is that there has been an increased HPV infection in the anal rectal junction, and perhaps this is one of the reasons why rectal cancer might be increasing. And finally, there might be a change in the microbiome. So in recognition of this increase, the National Cancer Institute is concerned to a degree where they've decided to set aside funding, as it is one of the now provocative questions which they offer. There's 10 provocative questions each year, and this is number one. What are the underlying causes of the unexplained rising incidence in certain early onset cancers? To find the evidence for a cause of an increase is very tricky. In epidemiology, what we would do, we would look to see whether the increased incidence is correlated with an increase in a risk factor. And this is an ecological analysis. And it is, the problem with ecological analyses are they're very weak. You can imagine that you can see correlations increasing with lots of risk factors, and it might not be the cause for the increase in the disease that we see. So what we need is a much more sophisticated research design. And this is why I'm pleased now to introduce my colleague, Associate Professor Dan Buchanan, who heads the Colorectal Cancer Oncogenomics Group, who has some great ideas on how to answer this question. So now I'm going to pass over to Dan. Stop sharing so Dan can share.
Hope that's working for everyone. Thanks, Mark, and thanks everyone for joining this morning. I'm going to pick straight off from from Mark's great introduction, and I really just wanted to reiterate that there is a big young onset colorectal cancer problem. We have tools that Mark has shown and that Mark's been involved with that can be very effective at reducing the development of colorectal cancer and decreasing its mortality in people over 50. But we don't have those same tools deployed at a national level for people under 50. And so this has in some ways resulted in this young onset colorectal cancer problem. So now between 10 and 12% of all new colorectal cancer diagnoses are in those people that are under 50. And this is projected to double by 2030. In Australia, colorectal cancer between 20 and 39 years of age is the third leading cause of cancer-associated deaths in this age group. And young onset colorectal cancer has quite a number of significant clinical challenges and social impacts. We know that the tumours that develop have more aggressive features. They're diagnosed at more advanced stage and often with metastatic disease when compared with later onset colorectal cancer. And alarmingly, it's been shown that the diagnosis of colorectal cancer in young adults is on average 6 months later than the symptom onset. And this reflects the fact that people aren't recognising the symptoms, both patients and clinicians, and many of those symptoms are often things that can occur outside of the scope of colorectal cancer, so those are often ignored, or and in some ways it can be related to being young and feeling a bit more invincible in terms of not taking much notice of those symptoms. The years of life lost can be high because of that early age of onset, and this can, if young onset colorectal cancer occurring in families means that it's typically occurring in families that have young children and things like that, so it can be quite devastating. From our survivors, and we've been able to engage with a great team of consumers and young onset colorectal cancer survivors through our research studies. They flagged that there's lots of ongoing issues and anxiety around their diagnosis, wanting to know what caused their colorectal cancer. Was it something that they did? Will it return? And what is that going to have an impact for the children? Will they end up developing cancer as well? So there's a number of significant issues here that we need to tackle. Mark's mentioned that there are a number of risk factors that have shown robust associations with young onset colorectal cancer, including obesity and physical activity, consumption of alcohol and red and processed meats. Inflammatory bowel disease, we know, is a risk factor for colorectal cancer in young adults, and having a first-degree relative with colorectal cancer increases your risk of developing colorectal cancer. And we know in the young people that around 25% of those young onset colorectal cancer cases also have a first-degree relative with colorectal cancer. So in terms of the genetics, around 20% of young onset cases have a pathogenic variant identified in a known hereditary colorectal cancer or polyposis susceptibility gene. And the majority of those, around half of those, are actually related to mutations in the mismatch repair genes and Lynch syndrome. What it means is around 80% of young onset cancers aren't explained by these known hereditary syndromes. And at this point, we really don't have a good idea of what is causing their cancer. And this is what is part of the big concern. We've recently been involved with some work published around using a 95 SNP polygenic risk score, which has been shown to be more strongly associated with younger onset colorectal cancer than later onset. And so it'll be interesting now as more work develops in this space to see how this interacts with some of these environmental, lifestyle and dietary risk factors. In terms of the histopathology of the tumours, we've mentioned that they can be associated with more aggressive features. They appear to be, there are a number of features that suggest that young onset colorectal cancers are a distinct group from the later onset. Mark's shown you previously that there's an increasing diagnosis in rectal cancer, and in fact, there's more distal and rectal cancer in the younger group than in the later group. And they're often diagnosed with synchronous metastatic disease. In terms of prognosis, there have been a number of studies suggesting that they might have worse or even better prognosis, but the jury's still a little bit out in that space. In terms of the molecular subtypes, the dominant subtype that is clinically applied is this, looking for the evidence of mismatch repair function. And mismatch repair deficiency is a significant proportion of the young onset group, and that's largely related to Lynch syndrome and mutations in the mismatch repair genes. But by far around the biggest group are those that have proficient mismatch repair function. This accounts typically for about anywhere to 80–90% of the young onset colorectal cancers. So by far and away the largest group. In terms of the somatic drivers, so BRAF V600E and APC mutations. So these are two mutations that are key drivers and key markers of different pathways of tumorigenesis. And both of these have been shown to be at a lower frequency in younger onset cancer than later onset colorectal cancer. On the flip side of that, then we do see a higher frequency of CTNNB1 and TP53 mutations in younger onset colorectal cancer. In terms of more studies looking at more omic approaches to younger onset colorectal cancer. We know that at least in the TCGA colonic adenocarcinoma and rectal adenocarcinoma studies, that young onset colorectal cancer is very underrepresented. So we really don't have a good and large-scale picture of what's happening in the genomic and methylomic space in young onset colorectal cancer. Again, similarly, the immune landscape in mismatch repair proficient young onset colorectal cancer has largely been unexplored. Some recent data suggests that even up to a third now of these proficient young onset cancers have immune features that they're associated with a high immunoscore, which may have implications for their treatment, and in a sense makes them look somewhat like mismatch repair deficient cancers in terms of their immune profile. The microbiome has been very topical in the last few years, and it certainly is, there certainly have been a lot of associations shown between the microbiome and colorectal cancer. But in particular, we're interested in three bacteria that have shown not just an association, but mechanistic evidence for their role in carcinogenesis, including the Bacteroides fragilis, Fusobacterium, and PKS E. coli. So we'll talk a little bit more about those later on. So we wanted to, I guess this is the paradigm that we're dealing with, and that to understand young onset or get to young onset colorectal cancer prevention, we need to identify who in the population is at risk of getting the disease. To be able to identify those people, we really need to have a better understanding of what's happening, what's causing the disease, and currently we don't know that. And if we can understand better the etiology of young onset colorectal cancer, we can tackle this problem of increasing incidence that Mark's talked about earlier. So that's where our research focus is at the moment, understanding the causes of young onset colorectal cancer, and then taking these and seeing if we can derive why the incidence is increasing. So our research aims, as I said, are largely around two scopes of work: trying to understand what the causes are and applying these four platforms of tumour profiling. So looking at the somatic mutation landscape in colorectal cancer, understanding what the microbiome or the bacteria-related landscape is for young onset colorectal cancer, looking at the DNA methylation changes that are occurring as part of tumorigenesis for young onset colorectal cancer. I'm not going to talk too much about the methylomic work today, but Eric Jewell is going to talk about this in a few weeks' time in his UMCCR presentation. And lastly, we want to understand what the immune landscape is. And so by looking at these four modalities individually, but also trying to understand how they interact to drive tumorigenesis in young onset colorectal cancer, we think will help us then identify features we can look at in pre-malignant lesions and back in the normal mucosa, which will help us get an understanding of who's at risk of developing the disease. So secondly then, by having this better understanding of the tumour biology, we want to see which of these, we want to know if the tumour biology changes over time. And if that's the case, that we might be able to assign some etiology to the increasing incidence. And we've defined this as, we're calling them temporal tumour signatures. And there's a concept that I want to bring up now that I would like people to have in their minds as we go through the next few slides, to look at the different results that we have for each of our platforms that we've been testing. And that's, we want to derive from tumour profiling patterns of mutations or changes in bacteria or particular signatures or phenotypes. And we want to be able to derive multiple independent and interactive signatures across a large group of young onset colorectal cancers that have been diagnosed over a long time span, in this case a 10-year time span, and see how these signatures might have changed over time. Then once we have identified signatures that have changed, we can then take that information and look at how dietary and lifestyle factors are associated with each of those particular temporal signatures. So we're very lucky to have two great resources of young onset colorectal cancer, which we're using to do all this wonderful tumour profiling. Firstly, our ANGEL studies, and these are obviously young onset colorectal cancer cases that are recruited from family cancer clinics across Australia. They're all mismatch repair proficient, and they've all had multi-gene panel testing to exclude known hereditary CRC and polyposis genes. So we're really dealing with that 80% of colorectal cancer that has an unknown etiology. We're collecting risk factor information, blood and tissue specimens, and to date we've got 125 young onset colorectal cancer cases recruited. The second resource, which is quite a remarkable resource, is the Colon Cancer Family Registry, and Mark is the PI of that. It began in 1997 and has quite amazingly had continuous NIH funding since that time, so it really is a fantastic resource. And to date we have 1,633 mismatch repair proficient young onset colorectal cancers that have been recruited through population-based registries in US, Canada and Australia. Again, we've been able to exclude known Lynch and bioleucemia TIH carriers. We have quite detailed risk factor questionnaire and biospecimens for each of these tumours. And you can see here the breakdown by sex across the 10-year period in which these young onset colorectal cancer cases were recruited, and here by tumour location. So I want to now show you some work in progress that we have going on in the lab to try and understand the biology of these young onset colorectal cancers. So today we've got 51 tumours that are all FFPE, 30 from ANGELS and 21 from the Australian Colon Cancer Family Registry. We've done whole exome sequencing, 16S sequencing, and in a subset we've done a 5-marker immune panel. You see the distribution of the ages by the two groups here, and by site for male and female in the colon. So by far and away, by the majority of the tumours that we're looking at are late-stage tumours. So these are the details of our methodology for whole exome sequencing. I'm not going to go through them, but I will say that this has been done in collaboration with AGRF, and we're very grateful for their collaboration in this in terms of the sequencing. So from our analysis, we're deriving not only mutations within the within genes, but also the microsatellite instability status, indel counts, tumour mutation burden, and of course we've developed pipelines to look at mutational signatures from this data. So really the mutational signatures are been around for 5 years or so, and been shown to they are the result of the accumulation of somatic mutations throughout the genome that in a lot of cases can be assigned to a specific etiology. And so we've applied those in the context of the known 95 mutational signatures in the COSMIC V3 reference, but also in this in terms of identifying a new mutational signature related to PKS E. coli. And we've done some work looking at de novo signature generation. So what does the somatic mutation landscape of mismatch repair proficient colorectal cancer look like? And this is work led by Dr. Khalid Mahmood. We see the majority of the mutations in our 51 tumours are missense mutations, and majority of those are related to C to T changes. Again, the majority of the tumours have a low mutation count, except for a couple of these tumours here. And you can see here the top 10 mutated genes in our cohort with the usual suspects APC, TP53, BRAF and KRAS. So what Khalid's done is taken the tumour mutation burden for each of these tumours and plotted them against other tumours from that have had large-scale genomic profiling done. And you can see our young onset tumour group here, which sits right next to our colonic adenocarcinoma group. And as you can see that there's not a great deal of difference in the tumour mutation burden between our young onset group and the chronic adenocarcinoma group. So this plot shows the top 25 genes mutated in our 51 tumours. Across the top here, you see the number of mutations for each cancer across here, and the top 25 genes here, and the proportion of, that they're mutated across, and the colour bars indicated here are the different mutation types. So you can start to see that when you get quite a heterogeneous picture of what somatic drivers occurring in the young onset tumour group. And so one thing we are considering while we're looking at this is how the patterns or the signatures of different and combinations of these somatic drivers be used to look for associations with dietary lifestyle factors, and remembering that this is what we think is underlying the majority of the causes of young onset colorectal cancer. So an example of that is this group of tumours here, which look to have somatic, the BRAF V600E somatic mutation plus mutations in TP53 and APC. And so that may be a group of tumours that we look at in terms of clustering, subsequently looking for temporal related changes in that cluster across a 10-year time period. So a second group of tumours that we might investigate down the track is ones that have KRAS mutations but don't have TP53 mutations. So you can see that there's different ways that we can, that we're looking to derive signatures or clustering or patterns from this data. So we're also looking at mutational signature analysis, as I've said, and this is a work led by Peter Georgeson. So Peter's done, calculated the mutational signature plots for each of our 51 tumours here. You can see, and this is using the set of known 95 signatures from the COSMIC set. I guess it's quite difficult here. It looks a bit like one of Antoni Gaudi's mosaics, but so it's a little bit hard to derive things in terms of what's happening from this picture itself. But if we look at perhaps a couple of signatures here, that this SBS signature, so single base substitution signature 2 here and single base substitution signature 3 here. So these are signatures that aren't typically or haven't previously been strongly associated with colorectal cancer. So that's something that is potentially interesting. And there's, you can see here, this blue signature here, SBS 39, is quite evident in a number of tumours here. This particular signature doesn't have a known etiology at this stage, but it's something that we could use, again, trying to understand that increasing incidence to see if, for example, this particular signature changes in time over this 10-year time period we're looking to assess. The other thing that Peter has done is look at novel new signature mutation detection. And so this is again under the premise that a lot of the international data sets have been underrepresented for young onset colorectal cancer, and therefore may not have picked up all the mutational signatures that are important or need to be or can be found within young onset colorectal cancer. And so Peter's done this and found 9 novel signatures in these 51 tumours. I've just highlighted a couple here that have quite a dominant set of signatures or signature within any one individual tumour here. And out of interest, one of the tumours here, we went back and looked at his, this person's risk factor questionnaire information, and interestingly, this person reported a history of inflammatory bowel disease. So that's something that is potentially interesting. We're going to follow up on as we start to accumulate more data. So this is just to show that Peter's applied the same thing, the same principles for 25 young onset colorectal cancer cases from the TCGA, and you can see the proportion of these novel signatures, 9 novel signatures that he's seen in the TCGA group. And again, we still see this SBS 39 signature here in a subset of the TCGA tumours. So moving on to our microbiome profiling in our young onset cancers. This is work led by Dr. Eric June. So this is the workflow that Eric's followed. And again, we're not looking at fecal or oral microbiome, we're looking specifically at the tumour microbiome. So he's used 16S sequencing in this pipeline here. He's looked at technical replicates from this data. And you can see here that for species that have a high relative abundance, the replication is good, but as the abundance gets lower, the replication is more variable. He's also assessed the alpha diversity between tumour and normal. So this is the variation in the bacterial composition within the tumour and the normal, and you can see that the tumour is significantly lower in its alpha diversity compared with the normal mucosa from these tumours. Looking at the tumour normal composition changes at the phyla level, Eric showed that in the normal mucosa here we have a decreased abundance of the firmicutes compared to the tumour, and here in the tumour a decreased amount of proteobacteria compared to the normal. So 16S is great, but it doesn't provide a resolution in some instances that we need to pick up particular strains that we're interested in, and one of those is the PKS E. coli. So Eric's designed some qPCR assays to detect this bacteria within our tumours, and we're specifically looking for this PKS island, which contains these CLBD genes, which produce Colibacter. So this is a toxin that we know induces DNA cross-linking and subsequent DNA damage, which has been shown to be a carcinogenic mechanism in tumour in colorectal cancer. This plot here shows the tumour and normal species level compositions for our 50-odd tumours and our matching normal mucosa samples. It looks a bit like our mutational signature plots. And again, at this level, it's a little bit difficult to derive what's going on, but just to highlight that there are some interesting levels of a relative abundance of different species in the tumour relative to some of these normals that, again, we can use to assess down the track as to whether some of these things are changing over time. Eric's also found that there are 4 bacteria that are more abundant in the young onset colorectal cancer tumours that haven't necessarily been associated with young onset colorectal cancer before. So we're going to do some, as part of our expanding testing, we're going to look at these particular species to see if this association holds up. I mentioned these 3 genotoxic bacteria that have established mechanisms in colorectal cancer, and Eric's assessed their prevalence across our young onset tumours so far. So for the PKS E. coli, we find them at 11% of the tumours, and both the ETBF and the fuso nucleatum we've seen it around 15% of the tumours. So it suggests that they're potentially involved with the etiology of young onset colorectal cancer, which is something we're following up on. I'll talk a little bit more about that in the next few slides. We're also looking at the tumour microenvironment to understand what the immune response is or what the immune contexture is in young onset colorectal cancer. And this is work led by Ryan Hutchinson and Julia Como in their lab. So this is the workflow that we've set up in terms of staining for CD3 and CD8, which is used to build an immunoscore for colorectal cancer. Ryan then annotates the different areas within the digitised slide, particularly looking at the tumour core and invasive margin, and then uses the software to be able to localise and quantify the cells within particular locations rather than describing a global density. And so this is basically the premise of the immunoscore, is that for a particular marker, say CD3, we look within the tumour core area to whether the expression is high or low, and we look within the invasive margin, again, to see whether the expression is high or low, and we do this for both CD3 and CD8. And if the expression is high in CD3 for the tumour core and the tumour margin, and for CD8 in the tumour core and the tumour margin, it's classified as having a high immunoscore, and vice versa. If the expression is low across these two regions for both markers, it's has a low immunoscore, and you can see the different scores in between that. So of course this has been shown to be particularly important for showing differences in disease-specific survival, where the high immunoscore group has the best survival and the low immunoscore has the worst survival. So we're applying that to our young onset colorectal cancer group, but we're also looking at a couple of other markers as well, which I won't have time to get into today. But these are CD68, FOXP3 and PD-L1. So looking at the immunoscore distribution in young onset colorectal cancer, of the 37 proficient tumours that we've tested so far, the majority are late stage 3 tumours. 13% had a high immunoscore, 54% were in the intermediate group, and 33% had low immunoscore. And you can see that the distribution by site within the colon. So again, just thinking about how we would take these patterns of immunoscore and then look at them as a temporal signature within young onset colorectal cancer, and then assess how dietary and lifestyle factors may influence the temporal immunoscore changes if they exist. So the other thing we're doing beyond just looking at CD3 and CD8 as part of an immunoscore is using multiplex immunohistochemistry and high-dimensional phenotyping to look at the expression of these in expression of different markers in the context of location within the tumour and stroma. So Ryan's been leading this work in collaboration with Ultiview and Definiens, and the idea is to build, again, what looks like a bit like a mutational signature plot, but in this case it's a correlation matrix for individual tumours based on different phenotypes that we can pull out of this multiplex immunohistochemistry assessment. So Ryan's applied this to mismatch repair deficient colorectal cancer in the context of Lynch syndrome to determine immune phenotypes that might be able to distinguish inherited versus sporadic mismatch repair deficient colorectal cancer. So they're looking now to, in collaboration with Ultiview, to move this into our young onset colorectal cancer study. So I mentioned we're interested in integrating these features that we're assessing, this tumour profiling, these different tumour profiling approaches. And so this is one example of how we're going about that. So this is the somatic mutation landscape that I showed you earlier that Khalid generated. These are key somatic drivers in colorectal cancer. And so we can see where each one of those has got a particular type of somatic mutation. And on the bottom here, we've got the different features that we can assess that against, including the age of diagnosis, starting from the very youngest here up to 50, the site in the colon, the stage of the tumour, the presence of PKS E. coli, Fusobacterium and Fusonucleatum, and B. Bacteroides fragilis, and whether these occur in the different in each of the 50 tumours that we've assessed, and of course then the immunoscore as well. So we can start to build up an interactive profile. For example, this tumour here has a high immunoscore, has a POLD1 somatic mutation, spice-like mutation in ATM, and a double hit in APC. Another tumour here has a low immunoscore. The tumour showed presence of both PKS E. coli and Fusobacterium, and has an RNF43 frameshift mutation and a BRAF V600E mutation, among others. And lastly, this tumour here that has a very high tumour mutation burden. We see a somatic POLE mutation, and we're just in the process of deriving the immunoscore and the rest of the bacterial presence within this tumour. So we can start to build up an interactive or integrative picture of what's happening in young onset colorectal cancer. So the last thing I wanted to touch on is this study that's published recently in Nature, which identified a mutational signature in colorectal cancer caused by genotoxic PKS E. coli. And so what they did was take normal colonic organoids and inject them with this PKS PKS bacteria here, and PKS with the CLBB gene deleted, and then looked at the mutation load across these organoids. And you can see that when the PKS was positive, meaning it's likely to produce colibactin, the number of single base substitutions was higher than when this PKS arm was deleted. And similarly, there was an increase in T deletions in the ulnar regions here. So what they were able to do is derive a specific mutational signature related to PKS DNA damage, which you can see here for single base substitutions, and you can see here for insertion deletion mutations. So this is really interesting. So it's a really interesting set of work that really draws that extra connection from association to mechanism in colorectal cancer. So we wanted to ask the question, does this PKS E. coli mutational signature cause young onset colorectal cancer? So Eric designed our qPCR assay to screen on our tumours, and we found it in 11% of our tumours. We looked for evidence of the SBS and the indel PKS unique mutational signature. You can see that we've seen it here in quite dominant at least one of these tumours, but also as a smaller signature set in a number of other tumours here. The indel signature is a little bit more prominent than the SBS signature, but nevertheless it shows that shows evidence that the DNA damage caused by colibactin from this PKS bacteria is causing or is having a role in tumorigenesis in these young onset colorectal cancers. And also we wanted to understand what the immune landscape was. So we've been able to assess 3 of these tumours so far that are PKS positive, and 2 of them were intermediate, and 1 of them was immunoscore low. And when we looked at the expression of FOXP3, it was high in the invasive margin region, and we didn't see any expression of PDL1. So really this is quite exciting for us. We're interested to develop this a bit further, and it really at this point is telling us that it does underlie an important subset of young onset colorectal cancer, and it is really defining an etiology for a subset of these mismatch repair proficient young onset colorectal cancers that were previously unexplained. So just to come back to this figure again, so a lot of the work I've shown you is trying to understand the etiology here and develop signatures from this tumour profiling. And so our idea related to the provocative question that Mark discussed early in the presentation was, how do we take this knowledge about etiology then to understand what's causing the increase in young onset colorectal cancer? And so this is how we are proposing to do it. This is our hypothesis and our plans that we want to do should we be funded. And it's basically to take the signatures that we're deriving that I've spent some time telling you about today. I didn't get a chance to talk about the methylation signatures, or the digital H&E histologic signatures that we're deriving as well. But to take her after these mutational signatures and then assess them for their change over a 10-year period. And so, for example, we might see that the PKS mutational signature increases in that time frame, whereas a DNA methylation signature may not. And then we can take those group of signatures that we know are changing and look for their association with specific risk factors that we've been able to collect as part of the structured questionnaire, and start to get an understanding of what dietary or lifestyle factors might be underlying this increase, so that as many of these are modifiable, we can start to target some of these to change behaviour. And lastly, to take this digital H&E and these quite novel and interesting deep learning approaches to see if we can derive a digital biomarker for some of these signatures that may be useful for future validation and a diagnostic screening tool. So in summary then, young onset colorectal cancer is increasing in incidence in Australia, but in most developed countries as well. It's likely to be mediated by dietary and lifestyle factors and not monogenic cancer syndromes. Now, current understanding of the etiologic mechanisms and drivers of tumorigenesis in young onset colorectal cancer is currently inadequate, and we can't affect any change currently with what we know. So we need new approaches and new ways to look at this process. So we've decided that we want to do tumour genomic, methylamic microbiome and immune profiling to derive novel sets of signatures that we can that might tell us something about etiology. And we're starting to integrate those, and obviously a good example of that is integrating our bacterial work with the mutational signature work to derive an etiology from perhaps 10% of young onset colorectal cancer at this stage. So our key findings so far, given that this is still work in progress, is that we see that there are existing signatures that haven't previously been seen in young onset colorectal cancer, but we're starting to see some of those existing in that case. And Peter's de novo signature analysis has a number of interesting novel signatures that we're going to follow up on. The 3 genotoxic gut bacteria show prevalence in between 10 to 15% in young onset colorectal cancer. And we've identified a number of other tumour-specific bacteria in young onset colorectal cancer. 13% of the young onset colorectal cancer were immunoscore high, suggesting they would have a good prognosis, while about a third were had immunoscore low. And we're going to apply additional markers and a multiplexed approach and high-dimensional computational pathology to generate integrative phenomic signatures for the immune contexture of young onset colorectal cancer. And this PKS coli mutational signature is present in young onset colorectal cancer. And while that may be a very basic statement, it really is very exciting to be able to say that for a subset of this group of 80% of colorectal cancers where we don't know what the etiology is, we can start to, for the first time, assign what we think is a non-genetic etiology to tumorigenesis here. So obviously our future work is related to our temporal tumour signatures, and if what we derive from our understanding of the tumour biology can be used to understand what's causing this increase in incidence of young onset colorectal cancer. So that just leaves me to thank a lot of people, particularly people in my group. It really is a fantastic group and a fantastic group effort, even though I've highlighted a number of postdocs that are leading the work. It is a group effort and a team effort to be able to pull these resources together and generate this data. We've got wonderful collaborators for the ANGEL study, the Colon Cancer Family Registry, and I mentioned the collaboration with the Australian Genome Research Facility, long-standing collaborations with Christoph Rosti, who's a pathologist in Brisbane, and of course Mark Jenkins and Ong Win and Finn McRae. And we're very lucky to have some fantastic consumer investigators who've been with us on this journey that we're trying to tackle young onset colorectal cancer. So I will finish there and I think hand back to Mark, is it?
Yeah, thanks, Dan. Dan, maybe you could go back to your slide with the overall design of your studies, which is about the third last slide, while I go to the questions. So first question we have is from Alex Basudis. And he asks, is it correct to say more young onset colorectal cancers are female, or is this an issue of selection volunteering? Is this a biological phenomenon? Well, Alex, I've just had a look at the latest incidence data and the counts. And although colorectal cancer is predominantly male overall, and younger ages, it's roughly even males, females, slightly higher for males. And the numbers in terms of recruitment that we have being predominantly female is somewhat due to females being more likely to participate in research. So that's the main difference there. Next question is from Sybil Sayer. She asked, Dan, in your introduction, you said APC mutations were underrepresented in young onset colorectal cancer, but that did not seem to be Khalid's findings. Dan?
Yeah, perhaps I didn't describe it properly, but they were less frequent compared to later onset colorectal cancer. So we know that APC mutations are a significant driver of colorectal cancer. And so the large study, I think it was from 18,000 or 36,000 colorectal cancers that were divided by under 50 and over 50, showed that APC mutations were less common in the younger group compared to the older group, but obviously clearly an important driver for both young onset and late onset colorectal cancer. And I think APC alone isn't the whole story.
Great, thanks, Dan. Bhamik Kumar Shah asks, are there any environmental carcinogens proposed for young onset colorectal cancer, Wi-Fi, etc.? Well, no, this is the major mystery for young onset colorectal cancer and the increase. The main risk factors for the early onset colorectal cancer are the same risk factors for older onset colorectal cancer. And this is work done by Oncowin that looked at that group specifically and didn't find any major differences. There could be some unusual differences, and you have to ask them these questions in a case control or cohort study to know. I guess the most novel one is perhaps HPV related through changing sexual practices in younger people over recent time, perhaps causing rectal cancer through HPV. But again, one study has looked at that specifically and didn't find strong evidence for that. So apart from that, no, we don't know. Alex has a second question. Are there large genomic catastrophes like chromothripsis, particularly in the P53 cases? I'm assuming these patients are stratified by family history. Dan?
Yeah, good question. We haven't seen any large-scale events like that. But also in saying that, we haven't necessarily looked for it. So it's a good question. And with the amount of TP53 mutations and their relationship with chromosomal instability and things like that, we really wanted to be in a position where we could go to whole genome sequencing for these tumours. But for various reasons, which would have helped to get a better picture of both chromosomal instability and chromothripsis, but we weren't able to do that. So we can certainly have a look, but I'm not sure how well we might be able to answer it.
Thanks, Dan. Another question from Sibel. She asks, are you controlling your observations across the omics with older age colorectal cancer? This is a great question, Sibel. We struggled with how to answer this question of cause over time, which is the increase over time. We struggled a lot with considering how to use the older age colorectal cancers in that question. Dan, maybe you want to summarise how we come up with what to do there.
We did. We flip-flopped quite a bit when we were thinking about this, and I guess it came down to two ways of thinking about it. One was to understand what the causes are, and the second was to understand what the increasing incidence is. And when we wanted to understand what the increasing incidence is, we needed to look at it temporally and not look at whether it was different from later onset colorectal cancer. So that's why we've gone down that path. But having said that, there is some with what we find with young onset cancer, it'll be important to show how relevant and translatable it is to later onset colorectal cancer. So I don't think it's something we won't explore in the future. It's just something we've been very specific towards for the provocative question.
Thanks, Dan. We've got an anonymous question here. Very impressive amount of work and great to see that this is being addressed. That wasn't from me, by the way. That was from someone else, actually. But a couple of questions. So immunoscore and phenotypes. Have you also looked at the consensus molecular subtype signature? Percentages of subtypes similar to what is found in older colorectal cancer patients? Would the young onset colorectal cancer tumours match the stromal subtype? So 3 sub-questions there.
Yeah, good question around the consensus molecular subtype. We haven't looked at that. We know I think one study has shown that CMS1 is the most common type in young onset colorectal cancer. We are thinking about ways we can derive that, the consensus molecular subtype, and one of those is by taking the methylation data that I didn't get a chance to talk about today and deriving the CMS from our tumour DNA methylation profiling, which other people have done before. So no, but hopefully we will, is the short answer.
Okay. And a question here about treatment. Are there different treatment guidelines for younger patients versus older patients? Do they respond similarly to therapies? I don't know the answer to that question. I'm an epidemiologist. Dan is a molecular geneticist. Dan, do you have any?
Again, I'm probably not the best person to answer, but I do know from the literature there's some suggestion that they might be treated more aggressively, and that a larger number of stage 2 tumours have adjuvant chemotherapy. But again, I'm probably not the best person to answer that.
Okay. I'm getting lots of questions coming in now, so I'm going to skip over people who have already asked a question previously. So we're going to go to Melody Grandin. Regarding the microbiome, do we know if there are any variations of the microbiome according to the seasons and subsequent food intake in humans, as it has been demonstrated in other species? If so, have you been able to sort this criteria in your study?
To me, the microbiome is a double-edged sword. It's fantastic. It is really important and clearly has a role in colorectal cancer. But there's so many variables that, including the ones you've just discussed, that we need to get a handle on to really take the microbiome forward to being able to use the information in treatment and management. So no, we haven't done any of that at this stage. But I think that's why that at least for what we've shown with PKS is that taking the presence of the bacteria and moving beyond that presence to show that it has caused DNA damage that can be seen within the tumour. I think that's a really important aspect that may help to alleviate some of the variability related to seasonal and different food intake and things like that.
Thanks. A question from Joette Vizes. If it seemed that there was a comparatively lower number of newly derived mutational signatures identified, why is this so? Did any of the newly derived signatures overlap with the established cosmic SBS mutational signatures?
Yeah, great question. Yeah, I think we found 9. In our first pass, we found 6. So clearly, the more samples we have, the more we're going to be able to find new mutation signatures. And it's a great question around the similarity between these new de novo signatures and the signatures that have already been identified. And Peter's had a look at that. And for the most part, I think the most part, they're less than 80% cosine similarity, so which isn't that great. So it's pretty exciting, I think, that the more samples we get, the more signatures we're now able to find, and the more we might be able to assign some of these to perhaps a dietary and lifestyle etiology.
I'm afraid we don't have any time for any more questions. I know there's lots of questions there, but you can contact Dan or me by email. Dan, do you want to present the last slide, which is next week's?
Yeah. So next week we have Professor Sue Evans, who's the director from the Victoria Cancer Registry, looking at fast-track notification of cancer to the Victorian Cancer Registry using ePath. So thanks everyone for joining today, and hope to see you, or hope that you can join next week for Sue's talk. And thanks, Mark.
Thank you, Dan. Thanks, everyone.